Hi, I am Helen Thomas. Here we are discussing the working principle of clinical chemistry analysis or biochemistry analysis. Biochemistry analyses are used in clinical labs to measure many analytes that are present in the blood or any other body fluids. It operates on photometric, electrochemical, or turbidimetric principles depending on the parameter that is being measured. The common methods that are employed in autoanalyzers are calorimetry, turbidimetry, ISC, enzymatic assays, and nephelometry. The principle of calorimetry is that it measures the intensity of the color formation by the chemical reaction. So it is based on the Beer-Lambert's law, which says that absorbance is directly proportional to the concentration. For example, the glucose estimation by GOD-POD method. Turbidimetry. So in turbidimetry, you measure the turbidity of the solution. So when the solution is turbid, there is a reduction in the light transmission due to the particle formation. For example, like immune complexes. It's also used in measuring the proteins like CRP or haptoglobulins, which also give a turbidic solution. ISCs or ion selective electrode are used in the estimation of sodium, potassium, chloride, or ionized calciums, which are having electric charges. So the principle is that there is an ion selective membrane, which is a semi-permeable membrane, and it is selective for the particular ion. Say if it is sodium, the sodium channel will be selective for only the sodium ions. It will only allow the sodium ions to pass through. So when the ion, say suppose a sodium ion passes through, there is a voltage formation and that there is a voltage change. And that voltage change is being measured, and that voltage change is directly proportional to the concentration of the particular analyte in the solution. Then we have enzymatic assays. The enzyme enzyme will catalyze a reaction and forms a measurable product. And say for example, urease will break urea into ammonia, and we measure ammonia, and by which we will understand how much of urease is there. Some high-end analyzers also use nephelometry, which is based on the principle of scattering of light at an angle from the particles that are present in the suspension. It could be employed for CRP, ASO, RA factors, or any other reactions where there is an agglutination of the particles. The general working principle of a biochemistry measure. First is the sample handling. So before that, if you have to prepare your sample, say for example, centrifuge it, separate the serum, or sometimes centrifuge it and give the mother tube as such. It depends upon the protocol and strictly adhere to the standard operating procedures. In sample handling, the machine will aspirate the sample and will add it to the reaction cuvette. Then there is reagent dispensing, where the reagent probes will be dispensing the reagents into the reaction cuvettes. Then there is a period of incubation and reaction, where the reaction will happen between the reagent and the analyte under the controlled temperatures in the reaction cuvettes. Then you have the optical measurement or detection, where a photometer will measure the absorbance at specific wavelengths. Then the results are calculated using the Beer-Lambert's law, and in the concentration is calculated using a calibration curve. The output is displayed on a screen or printed or is connected to the LIS interface. The key components of the analyzer are the sample and the reagent probes, the sample storage section and the reagent chambers, the cuvettes, then the photometer or optical sensors, the electrode system for IACs, and the software and LIS interface. Thank you.